Oh, shoot. Hey, ladies and gentlemen, welcome back. Didn't see you there. Scared me. Today, we are speaking with Dr. Armand M. Kuris, professor of ecology, evolution, and marine biology at the University of California, Santa Barbara, one of the foremost authorities on parasites and infectious agents. One of several decades, Dr. Kuris has transformed how scientists understand the ecological roles of parasites. As he said before we started talking, low hanging fruit. It was just easy to get in and say, I'll be the parasite guy. Your work ranges from fine grained natural history, sometimes beginning with the single snail, to global scale insights about fisheries, ocean health, et cetera, et cetera. It's good to have you on the program. How you doing today?
Well, thank you, Chris.
It's always a privilege to talk to people who are studying the environment, but as I can attest, all the people I've talked to, they're not too excited about the state of the environment. Is that something you resonate with?
It's a challenge, uh, but I don't predict future things. I don't know what the future will bring. Uh, Based on things that have happened in the past, it's unclear what will happen in the future. Right now, this is really largely a socio-political issue. It's not about the biology.
Hmm. Yeah, I tend to agree. I think that's a good answer. So you get into parasites. First, what's the most attractive part of it as far as your interest, or did you just go where it was needed, or did you have like you said? And what kind of parasites are we talking about?
Okay, well, there's a few things there, but just for
Do
history
what you
and to
can.
some degree amusement, I'm a fallen ichthyologist. I went off to grad school at Cal to be God's gift to minnow taxonomy. I had a very nice senior professor and fish ecologist, fish biologist. And after I was going to be his last graduate student, and after my first year, the next summer, he died of a heart attack.
Yeah.
And a good university, when these things happen, when somebody leaves for whatever reason, they work to figure out how to make an adjustment. But the fish guy was gone, and I had to do something else. The one course I'd had as an undergrad at Tulane University, which is actually a very infectious disease related university, being in New Orleans and all,
Yeah.
that the parasite course had fascinated me. I had no idea that it was interesting when I took it, but it had intellectually what I really like is complicated uh, topics.
Mm.
And bringing in host biology and the biology of the parasite almost inherently makes it more complicated than almost any other aspect of organismal biology
Really?
uh,
Hmm.
there. And so I decided to shift my master's, and I would work on these parasites of California freshwater fish for a master's. So I still had fish in aquaria. I was still out there catching fish. So the fish was still in my life. I did my master's. I got deeper and deeper into the intellectual aspects of parasite ecology and kind of left the fish behind. There's an aquarium back there over this shoulder and another aquarium over here. I still have fish in my life.
Yeah. Well, it's all interlinked, right? You're not just studying parasites; you're studying marine parasites, right?
Marine parasites, I did that for my thesis. But when I finished my PhD thesis, as I was writing it up and looking at where what would happen next in my life, I sat down for about half an hour thinking, who cares about what I was doing? And probably the phrase most associated with me, if you Google me, I bet you the phrase is parasitic castration. And so I apologize. So I had come to a realization that this was a specific thing. It was not just a nasty kind of parasite, but it was a distinctive kind of interaction, because the rest of the host isn't harmed in almost all the cases. Just the gonads are converted from host babies to ultimately parasite babies.
Wow.
And the
Wow.
host actually, as I increasingly began to think about this, actually lives longer than it does making fish or crab babies if the parasite is working the game just right.
This is like the cuckoo, but in the water.
Way more,
Wow.
way more. Cuckoo is a brief interaction,
Okay.
messing with one clutch. This is lifetime.
Ooh.
That's
Okay.
another aspect of parasitic castration. It
Huh.
is not temporary, it's for the rest of the host's life. The snails you mentioned, they, in our California estuaries, live 20 to 40 years. Many of them are infected in their second year. They're producing more parasite offspring than they could ever produce in terms of snail offspring.
Wow.
That's so. Anyway, that was the intellectual emergence from my PhD thesis that I had no idea when I started out that I would come to this sort of understanding of the concept. So I asked, who cares about this? And immediately that said, oh, this is about trematodes, because they all have snail first intermediate hosts. And the major trematode disease is human schistosomiasis. It affects about 250 million people a year today and kills about a quarter of a billion, a quarter of a million people rather a year.
Okay.
It's it's not malaria or tuberculosis. It's just
Sure.
the next step down.
Yeah.
It's a major tropical disease. So I said, oh, what I need is I'm going to seek a postdoc to work on this. I need to be able to interact with public health and medicine to bring this concept, because if these are blocking snail reproduction, there may be an option for biological control of the snails to reduce the medically important snails
Hmm.
there. So that was the initial concept. We're talking a 30-minute think back in 1971, 1970, 71. And so I apply for an NIH postdoc. I get the thing. And that when you get a postdoc where you have control of the money, the seller and the buyer shift in going forward because I'm bringing my own money. And so I interviewed people at UC Med Center in San Francisco, Johns Hopkins, University of Michigan, and went to UC San Francisco in part because, well, it was the most powerful of those organizations, but also UC and San Francisco. Okay, that's kind of my academic origin story.
Okay. And I have so many questions because the trematode, I've heard of nematodes. What's a trematode?
Flukes, they're flatworms. There's at least, well, about 20,000 described species. In the most important human disease are the blood flukes, the schistosomes, and there are three major species. There's still some work figuring out what exactly are species there, but Schistosoma mansoni is a major disease in sub-Saharan Africa. With enslavement of Africans, it was brought over to the New World and is a major problem in Brazil, some Caribbean islands, and Puerto Rico had a major problem back in the day. And then there's the urinary form, Schistosoma hematobium, which is also sub-Saharan Africa, but also the Middle East. Fortunately, that didn't come over. And then there's an Asian form, which has several species now, but it was originally described from Japan, where it was a very minor problem and resolved there. But China, still big problem in the Philippines, and then species in Southeast Asia, Vietnam, Laos, Cambodia,
Wow. So
Thailand.
this would indicate that they've been around for quite a while.
Oh yeah,
Yeah.
oh yeah. These are natural diseases. In fact, one of the thinking about human biology is one of the most important features in human biology is when we come out of Africa, because we are all African as origin for our origin story. We leave some of the worst parasites behind. We hardly ever pick up anything as bad as what we leave behind in Africa. It's one of the reasons we're so successful everywhere else.
The cold weather is not conducive to the more tropical kind of very wild ecosystems.
Some is climate, but it's also the hosts for these blood flukes. Depending on species, they have a vertebrate, and for the ones that we're talking about, mammalian, and for one of the human blood flukes, it's pretty strictly human. But they are very specific about kinds of freshwater snails, to the species, even selection for certain populations. The snails didn't leave Africa. And so we walk away from the life cycle of these things.
Are we in any particular trouble with some species? Obviously, you're talking about breeding at an impressive rate where they could overtake lots of biological beings. Is there any threats right now that we're supposed to be worried about? These little kinds of things taking out an entire species.
I don't know about taking out entire species, but climate change is moving boundaries. So to take malaria, for example. So malaria used to be much more worldwide than it is now. The USA was malarious well up the West Coast, central USA to southern Illinois, up as far north as New York. And when we controlled it, this was an American parasitic disease. When I arrived at UCSB, one of my mentors and ultimately the dean of research grew up poor in Oklahoma, and in the 1940s he had malaria as a child. That was an American disease. Well, now with climate change, we are moving some of these diseases north. So Europe was malarious, and England, Spain, France, Italy was very malarious. Some of the most important research, we don't usually think of Italians as great researchers. We think of them as great for many other things, but usually not science. Well, actually, early malaria research was some of the most substantial findings were done by Italian scientists because it was such a problem in the Po River Valley and other major areas in Italy there. Well, now the disease is moving north. We're also moving mosquitoes around. And so it's likely that these more northern places that, because of economics and social organization, were able to control malaria quite well, that they're more at risk.
Yeah. Or you could do.
There's another side to this. Every species actually has not only a low temperature threshold, but a high temperature threshold, and that is often met more abruptly. You fall off if the optimum temperature is, let's say, 80 degrees Fahrenheit. For a lot of things, when you're at 95, they're all going to die. But they could go all the way down to 35. It's not a normal curve. It's a curve with a sharp fall on the right side of the curve. So actually, there are some areas in Africa that may be less malarious because of conditions for the mosquito, for malaria, or the snails. So there are places where it's just not all a simple, it's always getting worse. There are
Mm.
some places where climate change, there's lots of other reasons to not wish another 5 degrees on Africa, but there are other consequences. So it's a complicated story.
Yeah. And we got to look out for the wet bulb in Bangladesh, and we got to look out for Jakarta will be underwater here pretty
Exactly.
soon, one of the most popular cities. So you talked about global warming. How about the pollution? Is there any showing that your research or something that we're seeing increases in these kinds of events with pollution?
Well, I've done some work with Kevin Lafferty, who I've collaborated with very extensively. But actually, when you look at pollution, again, for parasitic diseases, which most of them have 2 or 3 hosts in their life cycle, pollution often kills the snails or the crabs or the fish that are the intermediate hosts. So, in very polluted areas, lots of parasitic diseases fall out because one of the hosts collapses and there goes the life cycle. What
Mhm.
does increase are certain microbial diseases, bacterial or fungal, that actually can do very well in polluted environments. And so you are seeing some increases in some diseases, but you really have to go by what kind of disease it is to actually determine the impact on pollution. Overall, in a polluted world, parasitic diseases do worse.
Okay. So as we're killing ourselves, we're also killing those pesky diseases. That's a heck of a way to do it. Cut
Yeah,
off your
right.
nose to
Not the
spite
right
your
way.
face.
Not the
Cut off.
right way.
So you've also talked about, you know, parasite research that's directly impacted fisheries and conservation stuff. Can you tell me a little bit about this?
Sure. Well, I will make 2 points. One is when you're talking about fisheries, animal husbandry, or agriculture, the parasites are either they can be a big problem for you or they can be on your side. It depends on what you're trying to do. So, for instance, it's parasitic agents that are the main agents for biological control against insect pests. So the parasite is on your side, and the poor insect is going to pay the price for things you do to increase parasitism.
Hmm.
Uh, the Some of these exotic species, we did a fair bit of work on. One of the major invasive species is the European green crab. It was brought to North America on the East Coast well over 100 years ago. It showed up on the West Coast in the 1980s. It showed up in South Australia and Tasmania at about that time. It's in Japan now. It's in South Africa, spread around by shipping. And uh, in all of these places, and we did extensive parasitological research. We investigated the parasites. It's the best known crab in Europe, by the way,
Okay.
best studied crab, common crab. We studied their parasites from Norway above the Arctic Circle to Gibraltar. That's the entire coast of Europe. And many more parasites. In most of these other places, they were unparasitized. We studied them in South Africa, Japan, Tasmania, East Coast of the USA, which had a couple of things there, West Coast of the USA, where they picked up a parasite.
Hmm.
And we were evaluating them, and they have 2 parasitic castrators, by the way, that are perhaps host specific for that, if not the green crab and its close relatives it has no close relatives on the west coast of North America, for instance.
Okay.
And so there we detected possible control agents. To ever use them, you have to actually investigate how effective they are, and we had some information on that. But also, you need to be sure experimentally that they're safe in the introduced environment. And so it turns out that culturally, the places like Australia, which is very averse to anything exotic coming in,
Right.
they recognize they've done a lot of damage there.
The cane toad is one of the.
That's just one great example.
Right.
Yes. So the stakeholders are the ones who determine, are they willing to do this, promote this research, engage in control of the invasive pest. In agriculture, the answer was always, until the late 1990s, yes. Now they're much more circumspect about it because to some degree they have more pesticides that they can throw at it. That's part of the trade-offs that you get there. And so in the case of dealing with the green crabs, the parasites could be on your side. If on the other hand you're culturing salmon in pens, infectious diseases with that high host density that a pen would get are your problem.
Yeah.
So you really depends on what side you're on as to whether parasites are good or bad.
Yeah. Well, I myself, I think you nailed something that I'm really keen on, which is the zoonotic diseases and everything that comes from just putting animals too close together for food purposes.
Mm-hmm.
Not a big fan of that. So you did the snail. What emerging marine diseases and parasitic threats do you consider most urgent? I know I asked you this already, but I'm also, I know that there's much more than you're probably telling me that's going on in the ocean.
Well, we can talk about corals, for instance,
Okay.
and they have I think, a specific subset of diseases. Most of the coral diseases that are happening have actually been occurring in habitats that are, I'll say, marginal for coral. For instance, Florida. It's at the northern edge of the Caribbean. And so they have had a host of various fungal bacterial diseases that have been contributing, along with the climate change and the coral bleaching and other issues, have been making Florida, have been devastating what reefs they have in Florida. These sorts of diseases have not proven to be a problem. The bleaching issue is globally, I would say, at this point.
Okay.
But the infectious diseases, you're not hearing major stories on the Great Barrier Reef or in the Coral Triangle up in northern Indonesia, and the Solomons are. Uh, they they are in marginal areas, disturbed areas, uh, where they're building ports and other sorts of things like that, where there's a lot of uh, pollutant runoff. So it tends to be rather specific, associated with other things. And the nature of most of those diseases appears to be what we have called sapronotic diseases. So this is a concept that we developed 2015, I think, was our key paper on this. For all infectious agents, whether you're a virus or a worm, there are two basic aspects for the ecology and evolution. For evolution, there's a trade-off. The host and the parasite are in some sort of evolutionary interaction where host defenses evolve, parasitic virulence evolves, and there's a back and forth coevolution.
Okay. Tug of war.
Yeah. For the ecology issue, for all infectious agents, and this is such a strong principle, it's been called a theorem in parasite ecology or epidemiology. There is some host density where the numbers of hosts is so low that the chance of a parasite, no matter what its transmission mechanism is the host is so low that the parasite its offspring cannot get to a new host at the same rate, and they fall out of the system.
Interesting.
Infections would still occur, but you can't get at least 1 for 1 in every generation of the parasite. It's a threshold of transmission. But there is a class of diseases, that's what our 2015 paper with Kevin Lafferty and Sana Sokolow was all about, is that there's a class of diseases where both of those basic principles cannot happen. And that is, and as a good example, which thank God it's a rare thing, is that brain-eating amoeba that whenever it shows up, arrives in a publicity-related event, and in the USA, it's maybe 5 times a year.
Okay.
What that is, to just use that example, it's an amoeba that is living in still water, in ponds. It's a sapronotic thing. It's living on dissolved organics or feeding on detritus down in the bottom of the pond. That's where it occurs. By accident, if somebody manages to snort them or get them in their eye, these rare events, you have to be in that habitat, you have to have some sort of opening. They can have a complete physiology inside you.
Oof.
And they destroy the brain, the eyes, the brain. And so if you think about it, so the person dies, but all of those millions of amoebae that are eating its brain, they are also in a dead end. They need to go back in the pond.
Yeah.
That's where they belong.
Or they're hoping that the body turns into a puddle that they can just lounge in and wait for the next guy.
Yeah, if amoebae have dreams, that's what they'd be dreaming. So, in other words, there's no human host density that is so low that the risk changes. The risk is still, if anybody goes into the pond and has some terrible luck, you're going to get it. So that's a rare disease. By the way, a common disease with tens of thousands of cases, increasing, by the way, is valley fever.
Never heard of it.
A fungal disease that affects in mostly California and Arizona 50 to 80,000 people a year, with mostly cases that resolve, sometimes lifelong infections, and sometimes very serious pathologies, and occasionally somebody dies. Pretty much any California person dealing with infectious diseases is ready to consider that it's valley fever, and most physicians should be aware of it, especially where it's a fungus that needs to be in the dust. Southern Central Valley. Increasing, there have been articles in the paper just this year of increasing in the Salinas Valley, for instance. And it's also in
Interesting.
Arizona. It also affects dogs. The fungus needs to be in the dust. Every human it infects is a dead end for the fungus and a serious physiological issue for the human. So there's no coevolution because the fungus never gets to go from human to human. The human, the human may evolve defenses if this is a very common
Right.
occurrence. I would not be shocked, and I believe this research has never been done, that if you were to examine the genetics of the humans that before the Europeans got here lived in the southern Central Valley, I wouldn't be surprised if they had some defenses against this. So there could be evolution, but not coevolution, because the fungus doesn't ever get back to try it again. It's offspring.
How much would you wager the proliferation of shipping and just the interconnected world we live in has really done us in?
Okay. Well, before we get to that, I just want to get back to the
Please.
corals.
Oh,
Most
sorry.
of those coral diseases appear to be sapronotic.
Okay.
The sapronotic, white crown disease of bats is almost certainly a sapronotic disease.
Okay.
There are others that are major diseases for wildlife and animals as well as humans. So it's actually, in fact, in our paper, we sampled statistically a subset of the hundreds of fungal diseases of humans, and it looks like 90% of our fungal diseases are sapronotic. In other words, nobody gets athlete's foot from having feet rubbed against feet. You didn't get it from somebody who was your friend.
That's good. Now I can just go crazy with the feet rubbing. Okay, good. Thanks for that.
So that's a class of diseases that needs a different kind of modeling approach to understand the dynamics and a different kind of control. It's only about controlling the environment. To get back to athlete's foot, really clean locker rooms, wash them down with alcohol or whatever.
Seems reasonable.
Yeah.
Sapronotic infectious disease caused by an organism that's able to live and reproduce in the soil or another abiotic environment.
Yeah.
There you go. Yeah, keep it clean. So you study parasites, but these can be fungal. These can be amoebas. Does E. coli fall under that? I've had both. I had a coli and amoebas when I went to Mexico. I had the hot pair.
The E. coli was probably sapronotic, but the amoeba was almost certainly human. A mammalian parasite. Let's say a human parasite in Mexico. There, the amoeba can only live in humans that you presumably got.
Okay, good to know. I was always wondered too, is you're talking about the tug of war between parasites and creatures of all kinds. Do have a lot of marine life beaten most of the parasites out there, or are there some that are really always susceptible? You mentioned the crabs. Who is impervious?
I don't think anything is inherently impervious. If you're rare, infectious diseases are probably not your problem. Again, that host density issue. The more common, and this has been shown for sea urchins, which can have big die-offs from bacterial infections, is there is a threshold of transmission where you just don't see these epidemics where you start messing with the sea urchin predators, the sea otters, let's say, on our coast, or the spiny lobsters on our coast, then the sea urchin numbers explode, the kelp forests get devastated, and what brings those urchins down is almost always an infectious bacterial disease. So it plays a role, but if you're rare, infectious disease is probably not your problem. Some of these diseases, again, getting back to my favorite parasitic castrators, what they are able to do is they are able to interact with basically the physiology, even the genetics of of the host reproductive system. And that can be quite specific. So they tend to be evolved for being quite host specific, often to species. So they don't easily get to another species, but they're very locked in on the host. They have the ability to find hosts that are rare. So some of these parasitic castrators are found in 0.1% of the hosts. There's some that can be found in 90% of the hosts, but they can hang in there better than most things. It's the other type of parasite that is very good at finding uncommon hosts are the group of parasites that are called parasitoids,
Right.
where the parasite basically eats the host inside out. And the reason they are often good is their searching stage is often the adult. So an adult, to get to the ones that are biological control agents, an adult wasp, a creature with a brain and sense organs and flying ability that is often better than the poor aphid that it's going to parasitize, are very efficient at finding rare hosts. That's why they can be so good for biological control. Because in agriculture, it's not that I want to have 0 aphids. I want to have few enough aphids that they don't affect the yield, which is what I am going to convert to dollars.
Yeah.
So having a control agent that can find rare things is frankly way more effective than a pesticide where you don't know how rare the problem is, the aphid is, and you're doing it in a more area-wide, much less sophisticated way.
Yeah. Well, it's like fishing with a shotgun. It's
Yeah.
not very prudent. Glyphosate's pretty much in everything now. So you talking about how these parasites can survive. You've studied them in hydrothermic vents. What did you find in the hydrothermal vents? These are harsh conditions.
Yep. So uh, the uh, the hydrothermal vents are ephemeral habitats. The earth opens up, there's volcanic activity, it's intense, it's quite localized, and then the geologic events cease or fall away for a period of time, and those habitats then disappear. The people who study the biology of all the tube worms and all the other wonderful things down at the hydrothermal vents have described a cycle of the event comes, there's some early settling creatures, the tube worms elaborate, the smoke, the hydrogen sulfide begins to be reduced, they're replaced by mussels, and after 20 years, there's a whole different environment there. So the briefness of the environment means it's a real challenge for infectious diseases. Infectious diseases don't do well if you're breaking up the environment all the time.
Sure. Like rototilling soil. Sure.
Yeah. Yeah, or, parasites, the perfect place for a parasite is an agricultural field, animal husbandry, where there's high host density permanently. So easy for transmission to occur in one of those chicken factory farm places. On the other hand, when the hosts then on the vents, the hosts disappear, and that parasite, by the way, has to get from one vent to another. So actually, the studies that we did there were parasites are a very small part of the biota at the vents. Really disturbed habitats are not conducive for parasitism. A vernal pool, as beautiful as it is, as full as it can be with fairy shrimp and other things, the fact that it's out there for just weeks or months makes there's some parasites that can be there, but they're not playing a major role in the dynamics.
It's very interesting because it's a lot like the humans who wandered from Africa all the way up to Greenland and thought, at least nobody's bothering me here. I'll set up camp here. It's kind of nice. So we talked about the crabs. Let's see. You were working on coral reef fish. Is this true? You had showed parental care reducing parasite-induced mortality. So what's going on? Are there social strategies or behavior going on that's kind of helping out, knock out the parasites?
So the lead person in this is Lexa Grutter. She was an undergraduate here at UCSB and was faculty member at the University of Queensland in Brisbane. She's God's gift to cleaner wrasses. She's really done amazing work there. And so it was the key finding was that cleaner wrasses, which they eat parasites. They eat the parasite. That's why the fish come up to them. They reduce an amazing number of parasites when the cleaner wrasses are removed. And again, Lexa's work at the Great Barrier Reef at Lizard Island removed them for several years from these patch reefs and had control patch reefs where the cleaner wrasses were there. After a few years, all the bigger fish that can move from patch reef to patch reef were no longer found on the reefs, on the patches without the cleaner wrasses. They really were, it's structuring the fish biota on the reef, and it's because they're removing so many significant parasites of the fishes. And so, again, for young fish coming into a place, the behavior of having parents that are guarding things, parents that are caring for things, actually reduces the infectious. Mostly these are blood-sucking little isopods, gnathiid isopods that are the real problem there. It's kind of like a water mosquito.
Interesting. I always think of roly-polies when you say.
Yeah, the isopod is a roly-poly. These things don't roll much, and they're blood feeders. The females and the larvae are blood feeders. The males, once they mature as males, the males actually can't feed. They're just there looking for females with big jaws to fight with other males. Typical male stuff.
Sounds familiar, vaguely. Yeah. Well, look, you'll have to come back and entertain me and educate me a little more. We ran out of time. Wonderful to talk to you. Do you have anything exciting coming up that you want to brag about, or?
Yes. We have discovered a kind of parasite just a couple of years ago that it's a copepod. So that's a major group of crustaceans, which are egg predators. That is, they go into of crabs. They go into the egg mass and they feed. They suck out some of the contents of the eggs. The female crabs are holding their eggs, brooding them, depending on species, weeks, months, even a year for the red king crab in Alaska. And these things are egg predators. And the interesting thing about this is that no nyctothoid copepod had ever been seen on the west coast of the Americas, north or south, ever until 3 years ago now. They show up on commercial crabs in the Santa Barbara area and found as far as at least San Diego. And they can become so numerous that they can eat all the eggs of some female crabs. Total mystery. Where'd they come from? We have a paper coming out in Ecology. This is undergraduate research, by the way, something we can do at UC Santa Barbara as well or better than anywhere. It's coming out in Ecology, where we figured out how it's transmitted, how it stays in the system, and the next phase we're still working on is to quantify the mortality of the eggs. So that's a
Wow.
cool thing that
You probably work.
Dr. Ford.
There's new technology you're working with too. That's gonna. It's probably more exciting to see stuff. How you use an AI in the field to do stuff like this, recognize patterns yet?
No, parasitology involves opening up the hosts. And it's really pretty hard to get the information without doing it. eDNA gets you something,
Okay.
but for these parasites, they rarely have enough specificity that when they get an eDNA sample, you get so many kinds of things related to, let's say, malaria there, that you know that there are nowhere near that many described species. You do not know if they're environmental contaminants from something off a ship. So the information that's coming in is filled with a lot of noise, statistical noise on that. Still, I think the most useful thing is to open up the hosts. Each of them is like a little jewel box, and you open up, and there's some kind of little surprise inside, or not. That's what I like about crabs. Okay.
What you need a microscope too? You have to get down in there and see.
Dissecting microscope. Yeah. Low
Yeah.
power.
Okay. You'll have to show me one day. I have to check it out.
Okay. Well, come on out here. Santa Barbara is very nice this time of year. It's freezing
I've been out
cold in
there.
the USA, and right outside my window, right above the computer here, it's a pleasant, chilly 65 degrees.
How far are you from the cliffs?
The cliffs?
Well, the entire
Oh,
beach is like a cliff.
from the ocean, 15-minute drive.
Yeah. That's one of my favorite things. When I was a hitchhiker, I used to hitchhike through California, and we would sleep on the cliffs.
Santa Barbara,
Yeah.
the mountains that I'm looking at right here are 4,000 feet, about 20 miles from the ocean as the crow flies, the peak of the mountains. So we have
Lucky guy.
we have a drop that is like the Front Range to Denver, 4,000 feet to sea level here.
Watch your step.
I'm a Santa Barbara patriot. I totally admit it.
Yeah. Where'd you come from originally? Where were you? Where were you born?
Born in the Bronx.
Ah, okay.
Immigrant parents.
Cool.
Tenements, streets.
Nice.
Ethnic clashes.
Stickball.
And finished up growing in New Orleans.
Okay. I also lived there. I lived there. I lived in the Bywater. I lived in the Upper Ninth Ward.
Oh, wow. Wow. Very good.
Yeah.
Yeah.
Yeah. That's my claim to fame. I got around playing music for years and years and stuff. Oh, yeah. And before I go, I'll also plug, which is very important, my book, An Animal's History of Humanity. It's about the exploitation of animals. And this book, it's got words in it, so that's good. And if people want it, you can go get it and you can put it on your shelf, and people will think you're smart. They'll think you're learned because you have a book on the shelf. Armand M. Kuris, wonderful human being, professor of ecology, evolution, marine biology at the University of California, Santa Barbara, and the world's foremost authorities on parasites and infectious agents. Thank you for stopping by. Stick around. We'll say goodbye, everybody. We'll talk after this.
Okay.
Take care, everybody.